We are very happy to start the first Brazilian symposium on controlled human infection. This is something very new for Brazil, and we are very excited that we could bring a constellation of international speakers, and some very important scientists also from Brazil to discuss this theme here. So I would like to first pass the word to Fernando Reis, the Pro-Rector of Science of UFMG. He's representing the president of UFMG, Dr. Sandra Goulart, and he's one of the supporters of this event.
Thank you, Elton. Good morning. On behalf of our president, Professor Sandra Almeida, it's my privilege to welcome you to the first symposium dedicated to controlled human infection studies in Brazil. It's coordinated by the Center for Vaccine Technology of UFMG, Universidade Federal de Minas Gerais, and by the University of Oxford. This collaboration started with a visit of our research team to the Oxford Vaccine Group back in January 2023. That was followed by an agreement and by two subsequent visits of Professors Pollard and Ferreira here. And they now have established a collaboration that hopefully will lead to development of new products and to new achievements by these two great university groups. By the way, the most important university-based groups in the development of new vaccines. Controlled human infection is the deliberate controlled infection of healthy subjects in order to accelerate the development of new vaccines and treatments. It follows rigorous ethical standards to safeguard patient safety, but because the full development of new vaccines is relatively recent in our country, and I must congratulate, by the way, the group represented by Professor Elton Santiago, who developed the COVID-19 SpinTech, and also the Butantan group that will be represented by Professors Kalaz and Khalil for the dengue vaccine Butantan review. Fantastic achievements. But actually, because this is new in our country, this is somehow scaring for some people involved in research regulation. And so it's extremely important to discuss the topic in a high-level symposium with world-class speakers that I'm sure will be giving you a very balanced, unbiased, and instructive view of this approach for research and development. So thank you, everyone, and welcome to UFMG.
Good morning, everyone. It's a really great pleasure to have you all here, and I want to thank you all for coming, particularly for the international speakers that we managed to bring to Brazil, to Minas Gerais. And on behalf of the University of Oxford, I would like to introduce Professor George Hollander, who is the head of the Department of Pediatrics.
Thank you very much. I'd like to extend the thank you to all of you for coming to a very exciting symposium that has representatives from almost all continents to discuss something that is extremely relevant and important for the way how we wish to go forward and to deliver vaccines in a very timely and effective fashion. So on behalf of the University of Oxford, thank you very much for being here, and also great thanks to the two organisers and their teams that made this first Brazilian symposium, which actually should be more a global symposium, possible. So thank you very much to Daniela Ferreira and to Helton Santiago for putting together such an exciting program. I think the next 2 days will also provide ample opportunities to discuss among the delegates the different aspects of what is being presented and what is being reviewed here in a open and unbiased fashion, as we just heard. But it will also provide opportunities to initiate new collaborations for the benefit of human challenge studies. So thank you very much for coming.
Before, I would like to make some acknowledgement to some very important help that we have. I would start thanking Daniela Ferreira and the University of Oxford for all the support in putting this meeting together. I couldn't do it by myself, of course, and Daniela was a great person to work with, has been a great friend and a great collaborator in this project. Also, me and Daniela couldn't do it if it was not for the help of Anna Terzian, Jacqueline, and Claire. Anna and Jacqueline is over there. Claire is in UK. And all the speakers have much contact with both of them, and without them, we couldn't make this happen. So thank you so much for your amazing help. In addition, I would like to thank Thiago. He's over there, and his team, Virginia, is over here. They were responsible for the visual identity of the symposium and the website, which is very beautiful. I think I love it. I don't know if so. Thiago, thank you so much for taking good care of our communication. And we also must thank all the sponsors. And I would start thanking HICVAC for a generous donation, and the support of HICVAC really put us forward to put this meeting together. But after HICVAC, also CT Vacinas jumped in and offered some important help. Then the UFMG itself, I went to Fernando, asking him to help with the transportation of the speakers. And he said, I can do it, we can do it more. So he offered some money, and he offered support to advertise the event and the institutional support. So it has been amazing. Thank you so much, Fernando. Also, we received an important grant from FAPEMIG, which is the state funding agency, and from One Day Sooner, Jake here and Dudu is here representing One Day Sooner, which also supported the meeting. So thank you so much. And at last but not least, the speakers. We are bringing the top of the world speakers on the theme, and many of them paid their own travel tickets, their hotels. So we are immensely in debt with all the speakers. We have speakers that traveled like almost 24 hours to be here. So thank you so much for coming to Brazil and sharing your knowledge, sharing your experience. This is very important for Brazil to change some of our stakeholders' understanding about CHIM. People, as Fernando said, people is afraid sometimes of what they don't know, and we want to show them that this is not a big deal. Like, it's common for everybody that understands what is going on. So thank you so much, and we hope we have a great time, and you have a great time while in Brazil. Enjoy our food. We have an amazing food. So, and we prepared very carefully nice dinners for all of you. Thank you so much.
And just to end our introductions, I would like to introduce Maria Bueno, who is the Director of Governmental Relations and Commerce of the British Consulate in Belo Horizonte. Maria. Maria, vamos te ler? So, now it's better, right? Ladies and gentlemen, esteemed colleagues and honored guests, good morning. It is a great honor to stand before you today as we gather for this important international symposium on human health, a collaboration that highlights the enduring partnership between the United Kingdom and the vibrant state of Minas Gerais, and more specifically the best university in Brazil, UFMG, for the last 3 years, right, if I'm not mistaken. The best federal university in Brazil for the last 3 years. So this highlights also the importance of Minas Gerais to the UK. This is why we hold the consulate here. In recent years, we have witnessed remarkable advancements in science and health, particularly in the field of immunization. These developments are not only crucial for safeguarding public health, but also crucial in our pursuit of a healthier future for all. The UK has long been a leader in medical research and innovation, and it is through collaboration with dynamic regions like Minas Gerais that we can share knowledge, resources, and expertise to tackle global health challenges. This symposium serves as a vital platform for researchers, healthcare professionals, and policymakers from both the UK and Brazil to come together to exchange ideas and explore new avenues for cooperation. By sharing our experiences and insights, we aim to enhance immunization strategies, improve healthcare, and ultimately save lives. I would like to extend my gratitude to all the participants, more specifically to Helton and also Daniela, to make this event possible. Your commitment to advancing health through collaboration is commendable and truly inspiring. As we embark on this journey together, let us remember that our collective efforts can lead to significant breakthroughs in immunization and public health. It is an honor also to the consulate in Belo Horizonte to be part of this history. So I believe Professor Daniela can tell it a little bit more specifically because she was involved before. But I would like to thank also Lucas Brown, who was the former consul in Belo Horizonte, and also Melanie Hopkins, our former deputy ambassador for the United Kingdom in Brazil, that have brought them together, Oxford and UFMG, to start this collaboration. So all you see here today and tomorrow is part of something that started about 2 years ago, right? 3 years ago. So it's an ongoing process. Cooperation, international cooperation, comes from friendship, from relationship. So I thank very, very much to all of you, and I hope you enjoy the symposium from this day on. Thank you.
Hello? Yeah, now it's working. So I would like to introduce our first speaker. It will be Lucas Oliveira. Lucas holds a Bachelor of Laws from Federal University of Ouro Preto, a Master's about Transplant Bioethics by PUC, Minas, and a PhD in Law in Reproductive Bioethics from the Federal University of Minas Gerais. And he has a period of research at the University of Birmingham, and he is currently an adjunct professor of civil law at the Federal University of Minas Gerais, and the president of Minas Gerais Association of Civil Law Professors. Lucas has been working on bioethics of CHIM since 2023. Since 2023, and he joined the CHIM, Brazilian CHIM group to try to approve our first CHIM protocol, which I'm going to talk right after him. So, Lucas, thank you so much for being here and for helping us in this journey, and the floor is yours.
Hello? All right, hello everyone. Greeting to my colleagues. My name is Lucas Oliveira, and Elton previously introduced myself. I'm a law professor at the Federal University of Minas Gerais. My main research field is biolaw and bioethics. I have started to research about human challenge studies in my postdoctoral research within a project called BioJusPen, Bioethics, Justice, and Pandemics, coordinated by Professor Darley Dallagnol, along with Professor Brunel and Professor Alcino, who will speak tomorrow. So I guess I'm the odd one out here. Also, I'd like to thank Professor Elton, who gave me the opportunity to empirically approach my research and to be part of the first attempt of approval of a controlled human infection model in Brazil, with many and many outstanding professionals, researchers in a project with the highest scientific standards. For the sake of clarity and to fulfill my time, allow me to read my presentation. So, first of all, let's establish an initial concept of what we are talking about. So the World Health Organization states that controlled human infection studies or human challenge studies involve the deliberate infection of health volunteers. Such studies can be valuable for testing vaccines. They can be substantially faster to conduct than vaccine field trials, in part because few participants need to be exposed to experimental vaccines in order to provide estimates of efficacy and safety. Such studies can be used to compare the efficacy of multiple vaccines candidates and thus select the most promising vaccines for larger studies. Well, in a very brief manner, it's not my main goal here, but we can say that on today's standard, there are some minimal ethical criteria for human challenge studies to occur. There must be generation of important scientific knowledge, impossibility of sexual would not be regarded on today's ethical, medical, and regulatory parameters to be human challenge studies. But despite of that, they were all relevant to the actual conceptual and scientific understanding of human challenge studies, and they all involved, in some extent, the intentional infection of human beings for scientific purposes. Well, so the idea of this presentation is to provide a general historical overview of human challenge studies, appointing their main landmarks for today's understanding of this practice. This story is surrounded by malpractice and unethical experiments, what seems to explain the fear and the prejudice around this practice. But these cases were important to the contemporary ethical approach to control human infection models. Well, the idea of human challenge trials is superimposed on the development of vaccination in the 18th century. Smallpox was one of the humankind's worst enemies. It was highly contagious and had a mortality rate as high as 80% among infants in the 19th century. During the 20th century alone, smallpox is believed to have killed somewhere between 300 and 500 million people. In one of the great success stories of medical history, intentional infection of a research subject enabled Edward Jenner to develop the first vaccine. In 1796, Jenner inoculated an 8-year-old boy named James Phipps, the son of his occasional gardener, with cowpox, a related but far less virulent virus, and then repeatedly tried to infect him with smallpox. Poor Phipps, as Jenner later referred to the boy, never came down with smallpox. Jenner reported that 6,000 other people were vaccinated, and then the far greater part of them were challenged with smallpox. While Jenner's experiment would likely be considered unethical by modern standards, his challenge study gave birth to the field of vaccinology, leading to the advent of vaccination in general and the vaccine that enabled the eradication of smallpox. Although Jenner is regarded as the developer of the first vaccine, variolation began much earlier in Asia and Eastern Mediterranean and was introduced to England and North America in the early 18th century. Variolation or inoculation means the prevention of a disease by injection of material believed to produce a mild infection and thus convey an attenuated risk of a more serious disease. Well, in the late 19th and early 20th century, additional early challenge experiments began to occur on a larger scale with increasing scientific rigor. Challenge studies investigating what would now be referred as vector-borne diseases, such as yellow fever, malaria, and dengue, were particularly prominent at the time. Famous early examples of such experiments include the failed attempts by Carlos Finlay to transmit yellow fever from symptomatic patients to healthy individuals in Cuba, and the successful transmission of malaria via infected mosquitoes in Italy by Battista Grassi. The latter study provided the first experimental evidence that malaria was transmitted to humans by mosquitoes. In 1898, the United States Army occupied Cuba. A medical team was sent in, with Walter Reed as its leader. In 1900, experiments by Reed demonstrated the transmission of yellow fever to health volunteers by injection of blood from confirmed cases and by mosquitoes fed on confirmed cases. Reed's research ultimately led to the development of methods to prevent infection by avoiding mosquitoes. Reed is frequently credited for establishing a prototype of informed consent for research because subjects in these yellow fever challenge studies were asked to sign a contract that outlined the expected risks of this research. The contract also entailed the payment of $100, which was doubled for those who contracted yellow fever. Although no research subjects in these initial experiments died, the majority contracted yellow fever, in some cases with severe symptoms. In 1918, 3 French physicians undertook a series of challenge studies concerning the Spanish influenza. Charles Cole and Charles Lebel took a expectoration very rich in various bacteria of a sick patient, passed it through a filter, and injected it into 2 subjects with, quote, goodwill. One fell ill and the other did not. A third microbiologist, René Dugeric, subjected himself to a higher level of risk. In a hospital, he took blood from 4 of the most gravely ill patients, one of whom had died, mixed it up, passed it through a filter, and then injected it under his own skin. He describes the states of the body and mind through which he then passed. It's important to notice that self-experimentation was deeply used as a means to understand diseases and to test medicines and vaccines throughout the history of human challenge studies. In 1927, the Nobel Prize in Medicine was awarded to an Austrian psychiatrist, Julius Wagner Joheg, for the discovery of malaria therapy that consists in intentional infection with malaria as a treatment for neurosyphilis. It became a routine treatment in many psychiatry hospitals, administered either by mosquito challenge or by direct injection of human blood infected with malaria. The use of this therapeutic malaria infection was widespread in Europe, North and South America, and India until the 1940s, when penicillin was discovered as an effective treatment for syphilis. In any case, the patients undergoing malaria therapy were effectively used as research subjects in human challenge studies that improved and highly improved scientific understanding of malaria. And many of the atrocities committed in the research programs of Germany during the World War II involved intentional infection with pathogens, include anthrax, chlamydia, cholera, malaria, plague, among others. These programs collectively involved thousands of victims, many of whom died as a result. Prisoners were violently forced to participate. Participation often involved uncontrolled infection with pathogens known to cause severe disease, and sometimes involved torture and murder of those infected. In the United States, contemporaneous war-related research also include the recruitment of prisoners for infection in challenge studies. Although these were conducted under slightly more humane conditions, the voluntariness of consent and the legitimacy of recruiting prisoners for research has since been called into question. Of particular relevance to current malaria challenge research is the American military research in Stateville Penitentiary. In the 1940s, the University of Chicago in Illinois and the US Army collaborated on challenge experiments to test malaria drugs in 400 Illinois prisoners. These experiments involve infection of prisoners with malaria, including later studies with resistant strains of malaria. Later, studies of sexually transmitted infections performed by American researchers in Guatemala involved intentional infection of vulnerable groups with pathogens without their knowledge or consent, and also involved deliberately withholding treatment for these infections. Let me just go back a little bit. Okay, so all these object events gave rise to the ethics of research with human beings and bioethics, which established the moral and legal basis for conducting scientific experimentation with human beings. Many documents emerged from this context, among which the Nuremberg Code in 1947, after Nazism, and the Belmont Report in 1978, after several unethical experiments, such as the Tuskegee case. Among the various ethical guidelines for research with human beings, the following stands out: the absolute necessity of the voluntary and informed consent of the human subject, the rational weighting of risks and benefits, the fairness in distribution the benefits of the research. After the World War II, and with new ethical directives, the challenge studies involved volunteers from the general population. These studies involved a careful explanation of risks, voluntary consent, and isolation when necessary to prevent third-party transmission. Since the 1980s, improvements in scientific techniques, as well as rigorous regulatory and ethical oversight, have supported the development of multiple research programs. Human challenge studies have been largely concentrated in developed countries, especially in the United States and the United Kingdom. Studies collectively enrolling tens of thousands of healthy volunteers have been safely conducted with very few serious or lasting harms reported among participants. Here I mention two examples. First, the Walter Reed Army Institute of Research in the United States, which has a long history of conducting human challenge studies. Since 1986, over 1,000 volunteers have been successfully infected with Plasmodium falciparum. Second, the Common Cold Unit in Salisbury, England, a well-known experimental quarantine and challenge unit for respiratory viruses. The unit was set up in 1946. Over 20,000 volunteers, all under the age of 55 years, came from all over the UK to take part in the studies at the Salisbury unit. They discovered the first human coronavirus, the common cold virus, and were the first to describe the clinical effects of interferons. With the ethical and scientific consolidation of human challenge studies, this methodology has spread to several countries, especially to developed ones, due to the need of large investments, cutting-edge scientific knowledge, trained researchers, and proper infrastructure. Human challenge tests have proven to be fundamental for the fast understanding of disease and pathogens and have contributed decisively to the development of vaccines, having demonstrated themselves to be a safe method. For example, rhinovirus, a cause of common cold, has been the pathogen associated with by far the greatest number of human challenge studies. There were at least 55 studies enrolling collectively more than 18,000 participants. So this table shows some of the contributions of human challenge studies in contemporary research. So, to conclude, although the history of controlled human infection studies is marked by dark moments, proper regulations allow this type of study models to be used effectively and ethically, enabling important scientific advances. Thank you very much.
Thank you so much, Lucas. We have time for 1 or 2 questions. So, because of the time, thank you so much. Yeah, we are trying to maintain an English time, but we are Brazilians. In Brazil, everything is on time with 15 minutes delay. So just hold on that. So I would like to ask for my presentation, please. A minha apresentação. So I'm going to talk about a little bit about the future perspectives of CHIM in Brazil. And the pandemic showed that Brazil has important bottlenecks in innovation of vaccines. And there is this team of the Academy Brazilian of Science that discussed what are the limitations of Brazil to innovate in bioscience. And it was listed here and that Brazil has limited infrastructure for production of clinical batches, limited experience with safety tests and regulatory issues, and limited interaction between research groups and private industry. It's not on this document, but I also listed that we have limited expertise or experience to accelerate clinical studies, especially because we don't use CHIM. Brazil has a very robust basic science, a very robust clinical science, but we have difficulty in transposing the basic knowledge to clinical studies. And to try to fill in the gap, UFMG has the Center for Vaccine Development, which is led by Dr. Ricardo Gazzinelli, and this is the administrative board. And our mission is to accelerate the development of vaccines, immunobiologicals, and diagnostic tests for human and veterinary diseases, contributing to the country's socioeconomic development. The CT Vacinas right now has 7 principal investigators and 14 associate investigators, and a team that sums up to 92 individuals, and Daniela is one of our associate investigators. And seeing the difficulties that Brazil has in innovating, the federal government is sponsoring the creation of the National Vaccine Center, which will be a building of 8,000 square meters, where we'll be able to close the whole pathway for a vaccine concept to vaccine trials. And this building will include a GMP facility of 600 square meters to produce clinical batches for clinical trials. And CT Vacinas worked with several vaccine platforms, from recombinant proteins, plasmids, non-replicating viruses, attenuated pathogens, and RNA vaccines to develop vaccines that are important to Brazil. And a very important vaccine that is teaching us the pathway to move a research from the basic science to the clinical trials is the SpinTech. SpinTech is a vaccine that CT Vacinas developed against coronavirus that is a chimeric protein that uses the RBD of the spike protein and the whole sequence of nucleocapsid. And this vaccine not only protects against the ancestral strain, but also to all the strains and variants of concern that we have tested so far, protecting against death and against lung pathology. And this is the first vaccine that was conceived in Brazil, had all the production technology developed in Brazil, and had its clinical trial designed by Brazilians to enter in a clinical, in a first in-human study here in Brazil. So it was this is our first volunteer of the trial, and this day was a big party here at UFMG with the presence of the Minister of Health and Technology. Here it's me and Ana Paula from CT Vacinas, the Director of Science and Technology from the Ministry, and here's the PI, and the Secretary of Health of State Health of Minas Gerais. But CT Vacinas has also other vaccines in the pipeline, and the next vaccine to enter in clinical trial probably will be a malaria vaccine that is a CSP-based PVAX malaria vaccine, which showed up to 80% of infection protection. So this level of protection is remarkable for malaria vivax, and this vaccine uses 3 different CSP from the main PVAX strains. So Brazil has some immediate interest in chim. One of them is hookworm, which I'm going to talk a little bit in the next slides, and also malaria vivax vaccine. Also, we have interested in a Zika chim because Zika is still a health problem here in Brazil, and we'll have a roundtable discussing this tomorrow. But also, we are developing vaccines against leishmaniasis, schistosomiasis, influenza, which would all benefit from chim trials if we had that in Brazil. It's important to notice that most of the chim studies are done by developed countries. UK and USA alone account for 76% of these studies. We have other 14% by other developed countries, and developing countries just make up of 10% of all the chim studies performed worldwide. And in Latin America, only Colombia has ever done a chim study. So we are being left behind, and we want to catch up with that. So Rodrigo Correa Oliveira, which unfortunately we lost in October of last year, he was very important in putting up a project to develop a hookworm human challenge model for development of vaccines in Brazil. This was a collaboration with Baylor, George Washington University, UFMG, CT Vacinas, and also Fiocruz. And it's a project that has been sponsored by Wellcome Trust. And the rationale is that hookworm is still a severe health problem worldwide, affecting 500 million people. In Brazil, the prevalence is 2.73%, which means 5 million Brazilians infected, a little bit over 5 million Brazilians infected. And the Mass Drug Administration, which is the program that WHO is trying to eradicate the disease, has been shown insufficient to do so. So a vaccine is an important mechanism to alleviate the suffering of people that in rural areas that don't have access to sewage or clean water. And why a CHIM? Well, first, to evaluate the efficacy of a candidate in endemic countries. A phase 3 study for a worm vaccine is impractical because helminth infections is a problem of rural areas where you don't have a structure to conduct safe clinical trials. It's very rare to have a structure to conduct a safe clinical trials. And also the size of a phase 3 study is too large to be done in small communities of 100 or 1,000 people. So it's complicated. And the CHIM will allow better use of human time and financial resources. So we put up this plan to, which is in 3 stages. One, the hookworm CHIM is not new. It has been developed in the 80s and surfaced again the interest with the hookworm CHIM in 2000 to use as a treatment for inflammatory diseases. So it has been reported 181 individuals that has been infected with hookworm. So we know a lot about the model, including the adverse events that it may cause and what has never been observed. In natural infections, the main problem of hookworm is the anemia that it causes, especially in children and in pregnant, and it has never been observed in any of the study participants. So we put up the plan. We thought we have a very safe model to start CHIM studies in Brazil. And our plans were to transfer the model from GWU to UFMG, which is a protocol that was approved by our local IRB here at UFMG. Then we would proceed to a dose escalation study that would be done in Brazilian previously exposed to tune the model here in Brazil. And then a part C where we would test 2 vaccine candidates against hookworm, one of them with some very nice results, which David Dimmer, which is part of this project, is going to show a little later. And we talked with Anvisa, which considered the proposal viable and did not consider regulating the pathogen, so we received green lights of them. They took the same understanding as EMA, in contrast with FDA that wants to regulate the pathogens. We received the green light from local ethics committee, which considered the proposal innovative and ethically viable and approved the project. But we have a huge step back with CONEPI that considered the study unethical and revoked the approval by the local IRB. And according to CONEPI, the reasons that the study was considered unethical is because it has the potential to cause harm to the study participant which cannot be mitigated. So in their report, they said that the infection may cause anemia, which we know. So we put a very strong plan to mitigate that, including regular evaluation of ferritin levels and hemoglobin levels, which they just ignored, and they didn't tell us what in the protocol failed to mitigate any risk of the study. And also they considered that there are alternative methods to test efficacy of the vaccine, disregarding all our arguments that CHIM could accelerate and provide a safe and efficient model to test the efficacy. And also, it was not in the report, but it was in an audience minutes that we had with them. They considered the study futile because you can solve the worm infections with universal access to sewage system. And so you don't need a vaccine to help with infections. So here we can see that CONEP not only did not understand the CHIM concept, but also had some problems in understanding the parasite biology. For example, one of their issues is that the infection causes eosinophilia, which is like expected for any helminth infection. So this tells us that this symposium is really important to create awareness to Brazilian population, to the Brazilian scientists, to the Brazilian stakeholders about what is going on, what is being done all over the world. We could see that the countries that innovate in vaccines, all of them use CHIM to accelerate the development of their vaccines. And Brazil is being left behind. And it's like, it's very sad that we still find this kind of understanding. But there is hope. We have this symposium. We are very glad that all of you are here. And here are some stats. Until yesterday morning, after my calculations, there was more enrollment. But until yesterday morning, we had 177 registries to the symposium, 83 people registered in person. We had 94 online, and we have 11 countries represented here watching the symposium, either online or some of our guest speakers. Brazilian participants was 134, 49 total institutions represented, 28 institutions from Brazil, and covering 9 Brazilian states. So people is gaining interest in the theme, and this is very important to help Brazil advance its medical innovation. So I'd like to acknowledge first the Team Brazil team, Rodrigo Oliveira, which was a great friend and was very important for this program and for this meeting. The idea of the meeting, it was a lunch that me, Daniela, and Rodrigo was talking about. Also, I thank Ana Terzian, Andrea, Brunello, Cássia, Flávia, Jaqueline, Lucas, and Simone, which are the people that are still holding this project together in Brazil. Also, I would like to thank UFMG and CT Vacinas, especially Ricardo Gazinelli, the EN CT Vacinas, the Comitê Gestor, Fernando Reis, which is our pro-rector of science of UFMG that supported this meeting a lot, and the George Washington University, David, Jeff, Larissa, and Guaciara, which are also collaborators of the project. And with that, I thank you so much for your attention, and I'm open to questions if there's any.
Thank you, Elton, for the presentation. I have a question about the National Center for Vaccines. Where is that going to be located?
You've been to this
here.
Begatec. Yeah, it's very close to Begatec. So it's like across the street of UFMG campus.
But federally funded, you said?
Yeah, we are still discussing about the funding, but we hope it will be federally funded.
Okay, thank you.
Primus.
Yeah, thank you for your presentation. I have just two questions.
Mm-hmm.
I think with respect to the experiences your team must have with the ethics committees and regulators. The first one is were they explicitly invited for a meeting like this to also be exposed to what others have done and even ask questions? And the reason why I ask this is that I think I will be presenting tomorrow, and so these are really critical stakeholders that need to be involved early on, even before the study begins rather. Like in Kenya, we had engagements for over 3 years with these people. And once they get it, they can really be good advocates for the work. So what you are going through is not a unique situation. You need to really engage with them over a long period of time and get advocates who really understand the science as well.
Mm-hmm.
Yeah, and even if it means even supporting them to even attend international meetings where they also get exposed more to this also, it can really be helpful. So I'm not sure if they are part of this symposium.
They were invited. I invited CONEPI, I invited the local IRB, I invited other health committees in the city and in Brazil. We had few registries from CONEPI members and also few registries from the local IRB. I was expecting more, but that's what we have. But the local IRB, they were very open to discuss. So they gave me space to discuss the project with them in multiple times. They even held a section just to discuss CHIM and the project. So that was very helpful, and I think that the local IRB really got the understanding about what is it about. Now, I tried to do the same interaction with CONEPI, and I was denied. They didn't want to. All they gave me was this single audience with them. Me and my group, we went there, we presented, and that's the result. Yeah, that's what we have for today.
Hi, Aldo.
Hey, God.
It seems to be this is going to be a continuous fight because no matter what, they are prepared to say no, and this is very clear from the justification that they gave that don't. I don't know, it seems to be that you need maybe some political action, or I don't know.
Maybe. But the one thing that happens with these committees is that they renew. So I hope that CONEPI will change a lot in the end of this month. And I hope we can have a fresh start. But thank you so much. Because of the time, we must move on. Thank you.
So I have a very important role in this conference, which is to keep all the Brazilians on time, and I'm going to do my best. So it is a great pleasure to introduce you, Professor Sir Andy Pollard, from the University of Oxford. I don't think many of you will need an introduction about Andy. Andy is an infectious disease pediatrician. He's a director of the Oxford Vaccine Group. Most of you will know Andy for his huge role at the global lead for the clinical development of the Oxford AstraZeneca vaccine. But I want to say that Andy has done much more for vaccinology than just developing one single vaccine. He has been involved in probably most, if not all, of the vaccines and vaccine policy change in the UK, and also huge role globally on policy for vaccination and vaccine development. He's one of the reasons I came to Oxford, because he's a giant in the field of human challenge infection models. And I think I'm right to say that today he's not going to talk to you about pandemic threats, but he's going to talk to you about the amount of huge and brilliant work he has done on the field of human infection challenge. Andy, the floor is yours.
Thank you very much for that kind introduction, Daniela. We will continue to pay you now that you've said that, so good. So I'm going to talk about typhoid and paratyphoid controlled human infection studies and how those relate to the recent development of typhoid vaccines, but also for paratyphoid vaccines. And just to set the scene, typhoid is a disease that's been certainly around for millennia, but it's relatively recently that we started to understand more about it. This was the first really detailed description of the clinical diagnosis of typhoid by William Jenner. And William Jenner in the 19th century was a very important figure because he was the physician to Prince Albert, who was Queen Victoria's husband. And Queen Victoria's husband, Albert, died of typhoid, and William Jenner was there to make the diagnosis because he was an expert on the disease. And you can see here that typhoid is a disease which actually is rather non-specific. It's very difficult to recognize, different from other febrile illnesses that occur. And we didn't actually know the cause of it until a little while after Jenner's observations on the diagnosis, when Eberth, who was the pathologist, first described the bacteria, which at that time was called Eberthella typhi, which is much easier than the current name for the bacterium, which is Salmonella enterica subspecies Enterica serovar typhi. That's the current name for the organism. It was much easier when we just stuck with the Eberthella typhi originally. But really this disease is a disease of young children and school-aged children in countries which have poor sanitation, inadequate access to clean water. And just to highlight that, this is a study that we did in Kathmandu in Nepal about 15 years ago. And in that study, we just took blood cultures from children who presented to the hospital with a febrile illness in the summer, which is during the typhoid season. And 15% of those children had typhoid growing in their blood. Now, the pediatrician said, well, they've just got a viral infection, and gave them some antibiotics and sent them away, because that's how viral infections are treated. But actually, 15% had typhoid in their blood, so it was the right decision to give them antibiotics. And we've done many studies following those up, but the reason why this is such a big problem is because of this very poor quality water. So this is the municipal water system supplied in Kathmandu, and of course in many other cities in South Asia. And one of the problems is here you can see the water pipe, which is the white pipe, sitting in a sewer. And that's probably okay as long as you can maintain the water pressure. But as soon as the water pressure drops, you get ingress of the sewage or the surrounding contaminated soil, if it's in the soil, into the water supply, contaminating the water. I'm told that that municipal supply tastes better than chlorinated water, so that's why people continue to drink it. But across South Asia, there are so many cities of 10, 20 million people where you can't now put in the engineering cheaply to give them clean water and adequate sanitation. That's going to take a lot of money and much time. So that's really why there's been a big focus on development of typhoid vaccines. But the first vaccines were actually developed for the military, and the very first by Almoth Wright, who you can see on the left here, was developed in 1896 as a heat-killed vaccine developed in the Boer War in South Africa to protect the troops, because so many of them were dying in the trenches from typhoid and typhus rather than from the military conflict. But that vaccine was very reactogenic, and the troops really didn't want to take it because it made them feel so unwell. And then David Harvey, during the First World War, had developed another vaccine which covered typhoid and paratyphoid, another killed vaccine. Also made people feel very ill, but it reduced the rates of typhoid in the First World War, and about 90% of troops were vaccinated by the end of the First World War. Both the British Army and the French Army also used these types of vaccines. More recently, there are two vaccines which have been available for travellers to endemic areas. There's a live attenuated vaccine, TY21A, which is taken orally. And then there's the polysaccharide vaccine. Polysaccharide, called VI, decorates the surface of typhoid bacteria and can be made into a vaccine just from the purified polysaccharide from the organism. And that's what's been used for travellers. And in fact, WHO said in 2008 that these vaccines should be used to try and prevent this disease in children in those areas. And here's the message in 2008 when WHO said, we've really got to do something about this. But actually, despite that very determined message from WHO, nothing happened. And the reason for that was that a new generation of vaccines had been developed in the 90s and tested in Vietnam here, a VI conjugate vaccine, so the similar technology to pneumococcal and Hib vaccines. And this type of vaccine was shown to be very effective. You can see over 90% efficacy. But the problem with it was that the development stalled, but all of the countries said, well, let's wait for this new generation. Why would we use the current generation of vaccines? And so nothing happened with typhoid control until an Indian company developed their own typhoid conjugate. But they couldn't get anyone to take notice of it because no one believed that this new vaccine was going to provide protection. And fortunately, at this point, I'm just having a bit of difficulty moving on, there we go. At this point, WHO said, well, let's look at other ways that we can build confidence in these typhoid vaccines, and suggested in a statement here that use of controlled human infection models might get these new vaccines over the line, provide confidence that they worked. And so back in 2011 in the Vaccine Centre in Oxford, we set up the model for typhoid, based on a similar model that had been used in the 1950s at the University of Maryland. And at that time, just as you said in your talk, this was used in prisoners in the Baltimore State Penitentiary. And it came to an end, really, those studies, because of a change in the view in the US that actually prisoners were a vulnerable population and they were being forced to take part in these studies. Some of the investigators at the time tell me that the prisoners really wanted to take part because they got much better hotel accommodation when they were taking part in a challenge study that was far better than the Baltimore State Penitentiary. So to do this study, the first step was to develop the strain, which we did a lot of work in process development and then making strains under GMP, so that when you administer them to the volunteer, you know exactly what you're giving them. You know they're not contaminated, you know the dose you've got, and you can prove that when you want to have a look at the results of your study. You don't want to have variability in the challenge agent. And the way that this challenge is done is the first step is the gastric acid is neutralized by asking the volunteers to drink bicarbonate. And here you can see 120 mils and some very happy students drinking some bicarbonate. Just tastes a bit salty when you drink it. And then we prepare the bacteria, which we've been storing in the freezer, and we make them up in a small volume of bicarbonate. We check the dose, as you can see on the right there, to see actually what's in the inoculum. And then, not quite so happy now, but these are the students now drinking some bicarbonate which has been contaminated with the bacteria, so with wild typhoid bacteria. Just as would happen if you were in one of those low-income settings, drinking contaminated water with the bacteria in it. The volunteers get paid the going rate for the minimum wage in the UK for the time that they commit to doing this. There's no danger money allowed with our ethics committees. This is purely just reimbursement for their involvement, and they have to spend many hours with us, which is why they get a reasonable amount of money. And then the volunteers who then have had the drink of the bacteria may or may not develop disease. So this is what may happen. So some individuals develop no infection at all. And this graph shows the black line is the temperature, so there's no change in the temperature. The green line there is the C-reactive protein, so the measure of inflammation in the blood. So this is an individual who wasn't infected. You can see on the bottom there's an orange square there, which is there's a little bit of pass-through of the bacteria into the feces on the day of challenge. This is a typical individual who develops infection. And here you can see around about day 7, the fever starts, so the black line goes up because of the temperature, the C-reactive protein goes up because of an inflammatory response. And you can now see in red there in the blocks that we can actually grow the bacteria in the blood of this individual. So they have a bacteremic typhoid infection at this point. And they also start shedding more bacteria in the stool, which is the orange blocks there, the squares. Now, interestingly, there are some individuals who have completely asymptomatic typhoid. This is a medical student who was actually doing his final exams on this day in Oxford, and he's got blood that is now growing typhoid, but he's got no symptoms. There's no temperature, there's no increase in the C-reactive protein. And of course, he passed his medical student exams because we have a very high quality of student in Oxford. And the attack rates that we see in this model, if you challenge with about 10,000 bacteria, you get about a 2/3 of the individuals will develop symptomatic or bacteremic typhoid. So with that, we're able now to, having set up a model, to test vaccines, and we've looked at many studies, about 500 volunteers over the last 10 years or so, but this is the pivotal one for this particular vaccine, which is the typhoid conjugate vaccine I mentioned. And so the volunteers are vaccinated either with the typhoid vaccine or a control, and then a month later they're challenged with the bacteria, and then we take lots of blood samples from them, and at the end of 2 weeks everyone gets treated with antibiotics. And here you can see the top line shows the control group with the 2/3 or so of volunteers infected, and the vaccinated group have protection. Not complete protection, but there is protection shown in the model. Now, the data from this challenge study here published in The Lancet were then used to understand further about correlates of protection, which I don't have time to go into today, but we have identified levels of antibody in the model which correlate with protection, and later on we validated those in the field, but I'm not going to cover that today. So it's another use of the challenge model, is to start giving some confidence about which antibodies and what level of antibodies are required to protect against the infection. We also have used the model to study the response to antibiotics. And if you have a susceptible strain, then using fluoroquinolones clears the fever and the bacteremia much faster than the other widely used oral agent, azithromycin. Unfortunately, in the field today, almost all strains are resistant to ciprofloxacin. So azithromycin is more commonly used, which does mean that children who are infected get better slower than they did when there were susceptible strains. The data were used by WHO both for the pre-qualification process as well as for the policy decision. If you look at the policy document, there's a very extensive section on the challenge model as the critical supporting data to give confidence for the vaccine to be used. And the recommendation from WHO was that all children under the age of 15 in settings where there is a high burden of typhoid should be vaccinated against the disease. Now, we felt from this that the countries really needed more information to help them and give confidence about rollout. And so we then set up studies in about 100,000 children to look at the impact of the vaccine in low-income settings. And we did these studies across Nepal, Malawi, and Bangladesh, working with investigators in those countries with a total of about 100,000 children. So that's taking the data directly from the model to work with those countries to implement the vaccine. And over a 2-year period, studying the impact of the vaccine in this randomized controlled trial, we found about 80% efficacy, and this is just the data from Nepal, showing an 80% reduction in typhoid in that population, and in Nepal there were about 20,000 children in the study. And so this finished in 2019. Now, the studies from the 3 different countries were published, which you can see here. And I think importantly, in Bangladesh, we had sufficient very young children to show that the sorts of levels of antibody we were achieving in the challenge model and in the field studies were the same in very young children. They also were protected. Because one of the difficulties with challenge studies is we're often doing them to try and ask a question in adults about a vaccine we're eventually going to give to children. And so this field study was very important because it confirmed that the immune responses that we were seeing in the challenge model in adults were the same as in young children, and they got the same level of protection as other age groups, as you can see, possibly can't see, but it's in the bit in red at the top, showing they all had around 80% protection. The other thing that we were able to do in the Bangladesh study, which we ran as a cluster randomized trial in about 70,000 children, was that there was no evidence of any herd immunity. And this was a surprise because we had seen a reduction in shedding in the stool in the challenge model, but in an urban slum, because there's lots of movement of children across different clusters in a cluster randomized trial, we couldn't demonstrate any herd immunity, which is a shame in a sense, because if there is herd immunity, you get much more out of a vaccine than if you're only getting direct protection. So with all of these studies and the support from funding from the Global Alliance for Vaccines and Immunization for countries, the rollout has been continuing, and there's a big acceleration going to happen between now and 2030 with countries starting to vaccinate, largely driven by the very rapid spread over the last 5 years of extremely resistant Salmonella typhi. And so this rollout has now reached about 60 million doses distributed to children, and these are just 2 of the examples of rollout over the last couple of years for this vaccine. So that's typhoid. But the Salmonella pipeline is actually quite extensive. There's non-typhoidal Salmonella, which are a major problem in sub-Saharan Africa. A lot of work going on there, and there's a challenge model being developed at Imperial College in London, which may help with the development of those vaccines, where again the feasibility of field trials has been in question, although I'm pleased to say there are some field trials underway at the moment for non-typhoidal Salmonella. But with our interest in the invasive typhoidal Salmonella, the other big gap has been paratyphoid, because although there were those heat-inactivated vaccines over the last century, those are no longer available and were not very well tolerated. And there are 2 approaches for typhoid vaccines which are being used at the moment. With paratyphoid vaccines. One is a conjugate vaccine based on the LPS on the surface of the bacteria, and the other is a live attenuated vaccine. And there is additional concerns about paratyphoid. First of all, it's about 1/3 of all of the enteric fever. So enteric fever is the disease caused by typhoid and paratyphoid. About 1/3 of the disease is caused by paratyphoid. So even though there are typhoid vaccines being rolled out, clinicians who don't have the diagnostic tests available are still seeing the same disease because they're seeing paratyphoid. It looks the same clinically. And so it reduces confidence in the current vaccine programs. The second is that in several areas where typhoid vaccines have been used, and here's an example from China, you see a big reduction in typhoid, but coincidentally there's an increase in paratyphoid. So there is a concern, and I'm not sure whether it's epidemiologically properly validated yet, that the implementation of typhoid vaccines may open up an ecological niche that allows paratyphoid to take off, which is an added reason for moving ahead with development of paratyphoid vaccines. So the old typhoid vaccine that's no longer available, not very many good studies, but this one suggested that there may have been some protection against paratyphoid. And so that gives some confidence that it may be possible to protect against this disease. So we, some years ago now, started work on around about 2015 in setting up a paratyphoid challenge model, modeling it entirely on the typhoid model that I've already described to you. And one of the main differences is that we found that we could get about 2/3 of people to develop infection with paratyphoid, but with fewer bacteria. We only needed 1,000 bacteria rather than the 10,000 that we'd used with typhoid. With this particular strain, and the strain is an isolate you can see here on the tree from Nepal about 15 years ago. So what was interesting in comparing the two models, and of course this doesn't necessarily mean a difference between typhoid and paratyphoid, it's just a difference between the two strains that we have, was that with paratyphoid, the volunteers who developed infection were much less symptomatic. And in fact, most of them, when we do these studies, the diagnosis is purely on blood culture. We don't have a diagnosis based on them developing a fever. So it's a much milder infection. And for the volunteers, that's great because they take part in a study and they get paid and they actually feel fine. For the typhoid ones, some of them felt quite unwell. But interestingly, despite the lower dose and the lack of symptoms, if you look in the blood, the number of bacteria are pretty much the same, whether you're challenging with typhoid or with paratyphoid. So there's something quite different about the inflammatory response to paratyphoid. Now, we already knew that typhoid vaccines, heat-killed vaccines, were able to induce some protection from those relatively poor studies in travelers. But we also asked the question, what if you re-challenge someone who's been previously infected? Does a past infection give you protection? And in this study, we saw that past infection reduced your risk by about 50%, giving some hope that a vaccine that's based on a whole organism, at least, is likely to give some protection. We haven't been able to pull out any correlates so far from these studies to know what it is that was inducing that protection. So today there are several approaches for paratyphoid vaccines. There's a live attenuated vaccine and conjugate vaccines, and we have just finished the first study of the live attenuated vaccine, which is from the University of Maryland, our long-term collaborators, CVD 1902, which is attenuated with some deletions in a couple of enzymes. And these studies started just over a year ago, and as I say, we've just broken the code for this, and the good news is this vaccine works really well. And so we have the first vaccine since the 20th century history that will protect against paratyphoid. But the other approach is the use of bivalent typhoid and paratyphoid conjugate vaccines, and these are being developed. There's a number of developers, but the two leading ones at the moment are Serum Institute of India and Biological E in collaboration with GSK Global Health Institute. And this is just, this slide just shows you the development plan for Serum Institute of India that they presented to WHO's Product Development Advisory Committee. And you can see here that they have completed a phase 1 trial. And in fact, in my lab in Oxford, we did all of the assays for this phase 1 trial and showed strong immune responses both to the typhoid and the paratyphoid component of the vaccine. And their plan is then to do an efficacy study using the challenge model in Oxford and then phase 3 studies in India. So how does all of this fit into licensure for paratyphoid? So with typhoid, the challenge model was really important supporting data, but actually because there were licensed vaccines, you could actually license the typhoid conjugate vaccine just by looking at the immunogenicity in comparison with some of the previously licensed polysaccharide vaccines. But with paratyphoid, we have no available licensed vaccine. So the question has been, how do you get from where we are today with some potential vaccines to something which actually could be deployed? And this has been discussed extensively at WHO, and the problem is that licensure through field trials, proving the vaccine works in the field, is considered not feasible. And the reason for that is, I think, is not actually a trial feasibility question. You need about 60 or 70,000 people in the trial, and that is possible to do in countries like Bangladesh. But it's actually a financial feasibility. No one is prepared to spend the money for a trial and for a vaccine, which may or may not work, and we wouldn't have any information about whether it works without a challenge model. And so the discussions at the WHO committee, PDVAC, has been that this is the approach that will be taken, or at least is being advised for licensure. And until the process has gone through, we don't know whether it will work. But the typhoid component in these bivalent vaccines can be licensed just in comparison of immunogenicity with the existing typhoid conjugate vaccine. So that bit's straightforward. But it's been agreed that the paratyphoid component could be licensed on the basis of efficacy in the challenge model. But then the immunogenicity in the challenge model then has to be bridged to immunogenicity in field studies, but obviously much smaller field studies to show that. In fact, you only need a few hundred in the field in order to bridge the immunogenicity across all ages. And then obviously you need large safety trials, maybe of about 3,000 individuals in field studies. So this has been discussed with WHO, it's been discussed with the Indian regulator, and this is the approach that we're now moving forwards on, and hopefully in the next few months we'll get the paratyphoid study underway for this vaccine. So I think this is my concluding slide. In the future, we'll certainly over the next 5 years see many tens of millions more children protected against typhoid as a result of the work on the CHIM. There is a question which I haven't addressed today about whether boosters will be needed, but a SAGE working group at WHO has been formed this summer and will start to meet to consider whether booster doses are needed. And in about a year from now, we'll know whether the bivalent typhoid, paratyphoid conjugate vaccines work. And we will also be moving forwards with the live attenuated vaccine, which has already shown protective efficacy in the challenge model. But I think the bottom line with all of this is we really need clean water everywhere, because that would remove the need for these vaccines or vaccines against all of the other enteric pathogens that we're all worrying about so much. So I'll just end there. Thanks all to the funders, all of those people who have taken part in the studies, and our amazing collaborators, particularly in South Asia and Africa, who have been involved in these studies, and the wonderful team at the Oxford Vaccine Group. Thank you.
Thanks so much, Angie. We have time for one question.
Great presentation. And I totally agree that it's very important to advance and make fast. And that's very important for public health. But one of the big arguments that we hear here against this controlled infection is that the population is being exploited by countries that have developed the vaccine and have intellectual property of those vaccines. And so that would be not our, well, for public health is our immediate interest, but in terms of economy, there is the other side of it. And so my question is, how you're dealing with this situation, because this will be something that will come up to the discussions very often.
Well, it's a good question. I'm not sure it applies particularly to these models, because we're working with Indian manufacturers, and in fact you might take the view that they're exploiting students in Oxford. by the challenge model. And it is true that it's much harder to run these challenge models in the low-income settings where there's a very different perception of the risk. But I think there is certainly a view that development from a high-income country developer to then only market in low-income countries could be seen as exploitative. But I don't think really that's how this game works. It's much more, we need people to take an interest in development, and then it's the commercial drivers are critical whether you're a low-income manufacturer or a high-income manufacturer, that you can only sell at the going rate in those countries. But I don't think for these particular products that's an issue, and if you're making vaccines in Brazil, then challenge models in Brazil are very relevant to your population.
And
Thanks very much, Andy. Just some clarification on the paratyphoid vaccine. Is the opinion of the PDVAC that a CHIM study efficacy in adults would be sufficient to even license the vaccine for kids? Yes. With the appropriate immunobridging studies?
Yeah, so I think the great thing about the bivalent vaccine is that, well, there's two different ways of looking at it, but one way of looking at it is it's a typhoid vaccine with some added benefits, and rather like a pneumococcal vaccine with added serotypes. We don't know whether those serotypes are protective, but they look like they work in the same way, and we've got some evidence from previous serotypes they work. If you have a combination of two conjugates, one of which is shown to have efficacy at least in a challenge model, and the other which is very similar to licensed products, it's a much easier decision from a regulatory perspective. I think the other bit that's worth just emphasizing is that one of the reasons I think for doing these studies in the UK and not in India is that in India adults are all immune. They've had multiple exposures to typhoid and paratyphoid, and they're not very good representatives of naive young children. So I think students or young people, when I say young, I mean under 50s, in Oxford are probably a better model of a child in a low-income setting than adults are. But I do think we learn a lot from studying adults in low-income settings, but it's just not answering the question about a naive individual.
That's great. Thank you so much, Andy, for this. We're still within the 15 minutes Brazilian tolerance of the time. So we're going to have a coffee break now, and I would like to ask everybody to come back to the room in 20 minutes so we can try to bring the program back on time. Thank you so much, everybody. So we're going to start now our second session, and we should have Professor Angela Melassian from the University of Oxford online. Angela is a colleague of mine. Hello, Angela. Angela is a colleague of mine. She is a physician, infectious disease physician, and has done a tremendous amount of work on malaria human challenge models, both in Oxford as well as in many countries in Africa, including Tanzania, the Gambia, and Burkina Faso, and Thailand. So it's a great pleasure, and Angela, the floor is yours.
Thank you so much, Daniela. Can you hear me?
Yes, we can.
You can hear me? Fantastic. I'm just going to share my screen and then I'll start. It says the host has disabled screen sharing. Could you enable it for me? I don't think you have my slides, so if you could let me share, that would be fantastic.
Try again, please.
It still says to say.
Yeah.
Oh, that seems to be working.
Has it worked now? Great.
Yes, yes, it has. Brilliant. Can you see that on the full screen?
Yes, we can.
Okay, fantastic. So thank you so much for the kind introduction, Daniela. I'm really grateful to the organising committee to allow me to speak today, and I'm very sorry I couldn't be there in person this time. I really hope there might be another opportunity in the near future to meet with you all. So I lead a clinical malaria vaccine research team at the University of Oxford, where I work in partnership with Professor Simon Draper's lab, where we use human challenge models with which to test the efficacy of our malaria vaccine candidates. Now, it's important to distinguish between natural immunity, which obviously arises after years and years of malaria exposure, and vaccine-induced immunity. And the CHMI model, the controlled human malaria infection model, provides the ideal controlled experimental environment that enables us to better interrogate and understand these underlying mechanisms of immunity to either natural exposure or vaccines. So I have to begin by setting the scene with malaria for those who may be less familiar with it. So malaria is a parasitic disease that sadly causes more than 250 million cases, clinical cases, and more than 600,000 deaths every single year. And sadly, the majority of these, more than 80%, are in young children under the age of 5 years. There are 6 species of the Plasmodium parasite that cause disease in humans. The 2 main ones are Plasmodium falciparum, which is the big killer in sub-Saharan Africa, and the second is Plasmodium vivax, which is actually the most geographically widespread malaria, has long been underestimated, can cause severe disease, and brings with it a huge disease burden due to its unique ability to relapse due to reactivation of dormant liver forms of the parasite. This malaria is prevalent in many parts of South America, including here in Brazil. Now, the first part of my talk is going to be on falciparum, the challenges we've done both here in Africa and what we've learned from those studies. The second part I'll devote to vivax, which may be of more interest and relevance to this audience. So just to take you quickly through the complex life cycle of this horrible beast. So it starts with the bite of a female Anopheles mosquito. She injects one of these wiggly looking eyelash things under the microscope, which make their way through the skin to the liver, where they replicate over a number of days into mature liver schizonts in the hepatocytes. After about a week, they then rupture out of those into the bloodstream and start to infect red blood cells. And once it starts that process of infection of red blood cells, a continuous, exponential and uncontrolled cycle of growth in the blood ensues. And it's this blood stage of the life cycle that was responsible for all the clinical symptoms, the morbidity and mortality that we see with this devastating disease. Now, some of the parasites form gametocytes. These then get taken up in the next blood meal of the mosquito. They form an oocyst on the surface of the midgut, and then from there, the sporozoites, the wiggly things, get released back to the salivary glands, and so the life cycle completes itself. Now, this is just to show you that there are three main ways of administering a malaria challenge. You can either do it by direct mosquito bite injection of those sporozoites or through a mosquito bite. You can directly inject those sporozoites that have been cryopreserved, and you can do that either subcutaneously or intravenously. Or you can directly infect the blood stage parasites, the parasitized red cells, directly into the bloodstream. And what you can see here is after infection of volunteers, you monitor them for development of clinical symptoms and also for development of parasitemia, which you measure by a PCR in the blood. And the right-hand graph shows you what you see in terms of growth after a blood stage challenge. Parasites grow about 10-fold every 48 hours. On the left side in purple, that's what you see with a sporozoite challenge, either injected or via mosquito bite. Here, nothing happens for a week while the parasites reside in the liver, but after a week, they burst out into the bloodstream and you get exactly the same growth as you see in the blood stage from that point. And from these PCR data of the parasitemia, you can calculate the parasite multiplication rate. And that's our primary readout of vaccine efficacy when we test our blood stage vaccines. Now, most volunteers are diagnosed at a very safe, all at a very safe and conservative level of about 10,000 parasites per mil here in the UK. At that threshold, about half of them have symptoms, half don't, and they all go on to receive highly effective antimalarials. So it's a very safe model. Now, as a lab, our group has spent years making vaccines against the blood stage of the parasite to try and induce blood stage immunity. And we've also spent a lot of time working up the development of a blood stage challenge model with which to assess those vaccines. And this just shows you the complex procedures that are required in order to be able to do this in a very safe and stringent manner. Preparation of the blood-infected inoculum has to happen in a derogated Category 3 facility under aseptic conditions, everything in fumigated hoods, using sterile single-use reagents, following authorized SOPs under QA oversight, and every step of that process has to be carefully documented for full traceability. Once the parasitized red cells are thawed, they're then washed, the red cells are counted, and they're then diluted in sterile saline and loaded into syringes, again, sterile syringes, ready for infection of our volunteers in the clinic facility. After the challenge, we always carry out a parasite viability assay, so we know exactly how many parasites we've given these volunteers, and we also confirm sterility of the inoculum on culture. Cartoon of what you might expect to see in the blood following a blood stage controlled human infection. So what you're seeing here is the parasite growth curves over time. For a naive, malaria-naive volunteer, for example, an unvaccinated control volunteer in a malaria vaccine trial, it's the red line you'd expect to see, the 10-fold growth, PMR of 10. However, if somebody has received, for example, a partially effective vaccine, you might see the line in purple or blue where there's a reduction in the growth rate corresponding to a 50% or perhaps even 80% efficacy. The solid green line shows you sterile protection. We've yet to see that in one of our vaccine trials. It would be wonderful. Or you can see what is control and clearance as designated by the dotted green line. And you'll hear more about that and the interesting parasite dynamics that are seen in Africa using these models. I think one of Melissa Kapulu's group in Kenya will be talking more about this and show you the exciting parasite growth dynamics they see in their setting. So the parasite growth rate, or what we call the PMR, is the primary readout for a blood stage challenge. But what is the best model? Is it the mosquito bite model or the blood stage model to assess our vaccines? And we thought it was important to be definitive on this. So we looked at PCR data from 15 unvaccinated control volunteers, shown there on the top left in red, who received a blood stage challenge, and compared those to 15 volunteers who had received a mosquito bite challenge. And from the parasitemia data there, we calculated the growth rate, the PMR, and the bottom graphs show you the confidence intervals around those individual PMRs for each person. And you can see those are really tight for the blood stage model, but much, much wider confidence intervals for the mosquito model, giving you much less confidence and certainty in the true value of those individual PMR values. And then when you look at the mean PMRs across the two groups, you can see that's very similar. They're growing tenfold, but the standard deviation in the blood stage model is much less, whereas the spread of the data is much greater in the mosquito bite model. So this would make seeing a vaccine effect, if it was partial, for example, much more tricky to identify. And so this gave us the confidence that the blood stage model was going to be the best model for our purposes. And this is the model that we have worked most with over the last few years in testing our blood stage vaccines. And so back in 2014, we used this blood stage model to assess the efficacy of what was the best vaccine available at the time, so was thought. Historically, blood stage vaccines have been very difficult and problematic due to lots of antigenic polymorphism. This one at the time was called AMA1. It's one of the surface proteins on the blood stage parasite involved in direct invasion into the erythrocyte. And in fact, just before this, it had been shown in a field trial in Africa to produce some strain-specific efficacy in children, which is shown by the bottom graph on the slide, although the overall efficacy against clinical malaria was nothing to speak of, with the very similar graphs of the vaccine arm versus the rabies control arm. And so what did it do in our hands, in the UK human blood stage model, having showed no protection in the field in young children? So this shows you the parasitemia graphs in our hands. So the red line is delineated for the control volunteers, and the blue, the vaccinated AMA1 volunteers. And those graphs are completely overlapping, almost identical. And when you look at the growth rates, the PMR at the bottom, again, these look almost identical. Our worst ever p-value of 0.99 in a human challenge study. This was a definitive result, and important to say that the strain of the AMA1 was exactly matched between the challenge and in the vaccine. So you couldn't blame any polymorphisms in the AMA1 sequence here for explaining a lack of effect. So this was a bit of a nail in the coffin for AMA1, but important to know, and therefore the field had to move on. Around the same time, our lab had identified an alternative blood stage antigen called RH5. Now, this was different to all the others in that it was the first blood stage antigen found to be highly conserved and susceptible to vaccine-induced polyclonal neutralizing antibody. And it was then found to have an essential interaction with a receptor on the surface of the human erythrocyte called basogen. In order to invade and survive and cause disease, it has to bind there. So any blockage of that interaction, for example, by vaccine-induced antibodies, would mean the parasite would simply not survive. It would die. And so all our work from that time was on a new generation of vaccines targeting this new blood stage antigen Rh5, where we were looking to induce antibodies to block that essential interaction to prevent ongoing infection and disease. And so we went on to demonstrate potent efficacy, first in a stringent heterologous challenge model in Aotus monkeys. And you can see here on the bottom, the parasitemia growth curves in the control animals in pink, which are growing very nicely. Then in the animals who received a protein version of Rh5 with an adjuvant in orange, and those who received a viral vector-based version of Rh5 in green. And you can see the different parasite growth dynamics there with a combination of some sterile immunity and also a lot of control and clearance. And you can see on the top Kaplan-Meier that the majority of the vaccinated animals did not meet the diagnostic threshold. And so we then went on and did a human challenge study with this same vaccine. And delightedly, we had a positive signal in the human challenge model also. Now, this shows you the PMR in the control, unvaccinated people in blue, the historical AMA1 vaccinees in green, and in orange is our Rh5 vaccinated volunteers. So the growth went from tenfold to about eightfold, which corresponded to about 20% reduction in the growth rate. And although this was modest, to say the least, no other blood stage vaccine had produced any effect on the growth rate before that time. So this was a really important finding and a real turning point for the field. This just shows you that reduction in growth rate equated to a couple of days in delay to diagnosis in these volunteers, but they were all ultimately diagnosed and treated. And so, because we had seen efficacy in both monkeys and humans with a spread of protection rather than sterility, this enabled us to do a correlates analysis. And so what I'm showing you here is something called a growth inhibition activity assay, so-called GIA. And this we found it to correlate with in vivo outcome, both in the monkeys and in the humans. Now, a GIA assay is a bit like a virus neutralizing assay. So you incubate your parasitized red cells with your antibodies of interest, let a 48-hour growth cycle ensue, and then you measure the growth inhibition activity, which is measured by production of lactate dehydrogenase from the parasites. And what you can see is that the GIA shown on the bottom x-axis of both these graphs correlated very well with in vivo outcome in the monkey model on the left, and also with the reduction in parasite growth in the blood in humans on the right. And we were able to identify and define a threshold of GIA from the monkey model above which all the animals were protected. So a level of 60%, if you got above that, it meant you had protection. The humans didn't quite get into that protected red box, but the relationship was very much the same. And so what you can see here is the GIA produced by all our different vaccine regimens over the years. So the red box here is the human challenge model I've just shown you, where we saw the first effect of a blood stage vaccine using Rh5. But what you can see here is by iteratively improving these vaccines, first moving from AMA1 to the Rh5 antigen, then changing the vaccine platform from viral vectors to protein, the way it's delivered, and then moving from UK malaria-naive adults to African children, we were finally able to not only match but exceed the level of vaccine-induced GIA that was associated with protection in the Aotus monkey model. And so for the first time, we were then able to ask the question, does this level of GIA actually predict protection against clinical malaria in the field? And so we were delighted to be able to report very recently on our first phase 2b efficacy trial of Rh5 in Burkina Faso. the headline is that this produced 55% efficacy measured at our 6-month primary endpoint. The Kaplan-Meier there shows you the rabies control groups in blue and the RH5 vaccinated groups that are shown in green and yellow. And so this was obviously a massive milestone for the field. It's the first time any standalone blood stage vaccine has shown efficacy against clinical malaria in the field. But the reason I'm showing this is that having identified the correlate of GIA from the challenge models, we can now say that this is actually predictive of efficacy in the field for the first time. Now, interestingly, the UK adult CHMI gave quite a modest reduction in the growth rate in the blood, as you will remember from a few slides before. So the actual challenge model had actually underestimated vaccine efficacy in these African children, but it did give a signal. And so what about CHMI studies in Africa? So there's an increasing appetite from African researchers to do these studies, which is fantastic. And there's a lot of experience already with the sporozoite infection models, where you inject into the skin or intravenously, especially by Melissa Kapulu's group at Kemry. They're real experts in this, and you'll hear a very exciting talk on the data they've accrued, I think a little bit later on today. But we, for the first time, supported one of the Tanzanian PIs, Dr. Ali Olotu, in performing the very first blood stage falciparum challenge in Africa. This was a small proof of concept study where we infected these healthy volunteers in the same way that we do in the UK, with a defined dose of infected red blood cells. But we gave this to 2 groups who had a slightly different prior malaria exposure and therefore a different prior level of malaria immunity, a low exposure group and a high exposure group. We didn't know what to expect when we infected them, so this was a really exciting study for us. They were monitored in an inpatient facility for 28 days in Bagamoyo. And this is what we found. So this shows you the parasitemia curves over time, a very different spectrum of growth compared to what we see in the UK. What you're looking at here is the low prior exposure cohort in terms of malaria immunity in light blue, and in dark blue is the higher exposure cohort. And for the low exposure cohort, all but one of them did meet diagnostic threshold, but in the high exposure cohort, less than half of them did. So more had either sterile protection or controlled and cleared their parasitemia. And all these volunteers reached higher levels of parasitemia than we see in the UK, 50 times higher. And interestingly, without the development of the clinical symptoms we're so used to seeing in our malaria malaria-naive volunteers. And the bottom graphs there compare for you the time to diagnosis in Tanzania and the UK. And what you can see there with the dotted red line, by 10 days, that's where virtually all the UK adults have come down and been treated already. Whereas in Tanzania, many of them were still going very strong at 2 weeks with no signs of infection. And so this was a great first proof of concept blood stage challenge model in Africa. We demonstrated it to be safe. What was interesting, though, is that this really informed us that we would probably need to choose a lower prior malaria exposure cohort to be able to test our vaccines for efficacy, to be able to see a true vaccine effect, given the prior levels of immunity. So I'll just pause there for a second before I embark on Vivax. That's a whistle-stop tour of falciparum. So Vivax has a number of unique challenges which make it very difficult and very hard to work with in the lab. I've already said it's widespread. It can cause severe disease in a way people didn't previously think it could. It has this unique ability to relapse because of these dormant forms of the parasite called hypnozoites in the liver. In order to get rid of those, you need primaquine, and that means that a large part of the population who need that treatment can't have it because they're G6PD deficient. You get earlier formation of gametocytes, which has implications for faster transmission compared to falciparum. And the real problem with this parasite is that, unlike falciparum, you cannot culture it for any length of time. It's very, very difficult, therefore, to work with it, to produce assays, to test your vaccines or drugs. And there's a real paucity of access to human challenge models, which has been a real problem for many years. So through a fantastic collaboration with the Mahidol Oxford Research Unit, in Bangkok, through a contact with a PI called Jetsaman Prachumsri. I think her team are here today and will be presenting later. We together formed this EC consortium where we set out to develop a new challenge model for Plasmodium vivax to be able to test our vaccines for the first time. And so cases of vivax were identified from the Thai research team in the field at a southern site called Songla. These patients were once diagnosed, their blood was fed to mosquitoes. As soon as the mosquitoes were deemed to be infectious and the blood that was donated from the volunteers had undergone stringent safety testing and noted to be negative for all possible human transmissible infections, the mosquitoes were flown over to the UK, to a human challenge unit in London, where we had 2 volunteers ready and waiting to be bitten and infected. They then received the challenge and then were monitored in Oxford for development of clinical symptoms and parasitemia over time. And then at a defined threshold, a 250 ml blood draw was taken from these participants before they were drug treated. This was passed through a leukodepletion filter and then frozen down for future use under strict GMP-like conditions. So we had a challenge agent banked and ready for use, and we sent this to the Sanger Institute, where Julian Rayner and his team sequenced it for us using a new hybrid assembly method. And this is actually the highest quality genome for Plasmodium vivax to date, and it's ready in our freezer. We have about 190 vials and ready for use, and we're very keen to provide this to any interested collaborators who are interested in doing blood stage challenges to be able to have access to this. I've jumped ahead of myself, sorry. This slide was just to tell you about the stringent testing that this new isolated challenge agent had to go through in terms of GMP testing for sterility, extensive bloodborne infections. We had to test for viability, prove we had viable parasites, get a sense of how many. But it was a successful process, and that's really transformed our ability to be able to test vaccines. But of course, the first step was to show that we could safely infect naive volunteers and show that they could develop parasitemia. So we did this in the UK in a small study where we gave it at 3 different dilutions. All the volunteers developed very robust parasitemia and also gametocytes. And since then, we've infected about 40 people with no safety concerns, all recovered with treatment. And so we were finally in a position to start testing our vaccines. So what do we know about Vivax that has helped initial vaccine design? Well, we know its widespread endemicity. We also know that a place where it seems apparently lacking is sub-Saharan Africa. And we know that the population here are negative for the Duppy blood group antigen. This association was identified by Lou Miller back in the 1970s. Then in the 1990s, Chetan Chitnis demonstrated a critical interaction between the Duppy blood group antigen on the surface of the reticulocyte and the so-called Duppy binding protein, similar to the critical interaction that we saw with RH5 and Basagin that I've just told you. But despite this identification of a clear target, it took 30 years before anybody made a vaccine targeting this antigen. And so back in 2017, 2 vaccines had completed phase 1 testing, 2 Vivax vaccines. One was a vaccine we made in Oxford, which was a viral vectored formulation, a ChAd prime followed by an MVA boost, incorporating region 2 of the Duppy binding protein. And a second was Chetan Chitnis's protein version of the vaccine administered in a strong adjuvant. Now, both were safe, induced high levels of antibodies, including strain transcending antibodies. But until we had banked that Thai isolate, there was no way of assessing them for vaccine efficacy. So this is what we went on to do. We were able to test these candidates head-to-head in a human challenge study. What I'm showing you here are the control data points. This is the PMR, the growth rate in control volunteers in orange. The viral vectored volunteers are shown in pink and purple here, with no change in the PMR that's significant. The protein version of the vaccine, given with the matrix M adjuvant, again, didn't show any difference. It was just like the controls, given in a 0-1-2 monthly regimen of 3 doses. What was really interesting was the same vaccine, the protein version, given when you gave the 3rd dose at a delayed 14-month time point. This was more delayed than we had hoped by the pandemic, by the way. It was supposed to be a 0-1-6 month regimen, not a 0-1-14. But interesting that that really delayed 3rd dose led to a significant reduction in the PMR, greater than we saw with falciparum with the RH5 vaccine. This was a 50% reduction. So fantastic to demonstrate that in the human challenge model. And so what immune measures were predictive of this in vivo effect? So I've told you that we can't culture Vivax for any length of time, but we have a great collaboration with Rob Moon at the London School, who spent many years developing transgenic parasite lines using another monkey malaria called nullsci that's very similar to Vivax and can be adapted to culture in human red cells. And so by using the CRISPR-Cas9 technology, the critical Duppy binding protein gene in our vaccine could replace the nullsci version in the transgenic parasite line. So this is what he did. And then in the assay of GIA against a panel of potent monoclonal antibodies against the Duppy binding protein, he was able to demonstrate high levels of GIA with a whole panel of monoclonal antibodies against that. And that's compared to a negative Ebola antibody control, which is the flat black line just here. And you didn't see any significant GIA when you looked at this in the wild type parasite. And so this shows you that the GIA assay on the bottom right there did actually correlate again with the in vivo growth inhibition in these volunteers. There was also a correlation between in vivo growth and the antibody levels and the binding inhibitory antibodies. Interestingly, no correlation with T cells. Just antibodies. But this was really important because this has really given us a clinical benchmark to help us define better improved Duppy binding protein vaccines going forward and perhaps even using better antigen targets using this Plasmodium nullsci transgenic parasite line in the GIA assay. So a huge amount of progress there. So in terms of next steps, we were lucky a few years ago to get funding from another EC, a grant called OptiVivax. This we had developed with 9 different industrial and academic partners shown on the slide there, where we once again were able to bring Plasmodium vivax to the forefront and take our learnings from what we learned from the blood stage model and apply this to making vaccines against the other important stages of the life cycle, especially so for vivax, so the sporozoite stages and transmission stages. And we set out to make better vaccines, to identify new antigen targets, use new tools, new functional assays, and in addition, set up, a little bit controversially, the first ever relapse model for Plasmodium vivax. And this is what we're particularly excited about now. And so I'll just finish with 1 minute on this new study that we are in the process of preparing. So the idea here is that we use our Vivax clone, the agent that we banked in Thailand. We send this to the Netherlands, to our collaborators at the RUMC, who are experts in mosquito challenges and blood stage. They will infect healthy volunteers with this blood stage inoculum intravenously. They will then maximize the infectivity of that to mosquitoes by treating with gametocyte-sparing drugs. And then once the mosquitoes are deemed to be infectious, having fed on the volunteer's blood, those mosquitoes will then be transported back to infect our healthy and willing volunteers in the UK. And essentially, these will be 5 people. It's a proof of concept study. They'll receive a Vivax sporozoite challenge by mosquito bite. They will then receive treatment for the blood stage of infection, but they won't get primaquine. So we want them to relapse, and we expect them to relapse maybe 2 to 3 times over a 6-month period. Then at the end of the study, they will get the definitive treatment with primaquine, and then we'll continue for safety reasons to follow them up out to 5 years. So here we're looking at the safety of the model, the feasibility of the model, the frequency of relapse and time to relapse, and try and get us a better understanding of the immune response to Vivax, and really set up a model that can be used in the future to test drugs and vaccines. So I'm sure I've run over, so I'll leave it there just to say a massive thank you to everybody in the lab back in Oxford, our many, many fantastic collaborators, our funders, and our wonderful volunteers who we subject these challenge agents to many, many times over. We couldn't do this without you. And thank you very much for your attention, for listening. And I'm very happy to take any questions. Thank you very much.
Thank you so much, Angela, for this excellent talk. We are running a bit late for time, but we have time for one question, if there is any pressing question. Angie?
Angela, thanks very much for that very nice presentation. I just wondered if you could give some overview of where Malaria Challenge sits in vaccine development for malaria. Is it an essential component? Can people go directly to the field to develop malaria vaccines without going through Challenge? And is it, perhaps as appears to me, that it's a gatekeeper for what can go into the field?
I think it does remain a gatekeeper. There's definitely a role for it, and you can see how much we've learned from this. And we, for the first time, have shown a vaccine now to have clinical efficacy against malaria in the field that did have a signal in our challenge model, albeit a small signal. I think we can fairly definitively say that any vaccine that has shown to have no effect in a challenge model will not have an effect in the field. That's pretty clear in terms of the experience that has been had. So I think it's a good gatekeeper for ruling something out and saying there's no point in taking this forward. What you don't want to do is ignore a small signal, because that could actually mean something fantastic, as we have just shown for the RH5 vaccine. So I think there's definitely a role. There's a big debate on whether challenges should be done in the UK or in Africa, or in endemic countries for Vivax, such as Brazil, rather than in the UK. Historically, it's been much easier to set up models in the UK and the US, and I think there is still a value in doing that, especially if you're looking at a truly naive population. So depending on the question you're answering, you want to answer, I think there's still a role for the naive population outside of the endemic areas. But the endemic area CHMI, I think, is the future, and it's gone in leaps and bounds in Africa in the last few years. It's always more difficult in resource poor settings, and you need the ethical and the regulatory infrastructure in place. But there are many places now showing a lot of promise, and we have a massive expert in the audience, Melissa Kapulu and her group, who will be able to show you the exciting data they've accrued and the implications for product development on sporozoite vaccines. So yeah, I hope that sort of answers your question.
That was great. Thank you so much, Angela. Thanks so much for joining us online. And we're going to move on now to our next speaker. So it's my pleasure to introduce Dr. Wanlapa Rubsong from Mahidol Vivax Research Unit. We're going to carry on talking a little bit more about malaria Vivax.
Good morning. My name is Wanlapa Rubsong from Mahidol Vivax Research Unit, or MVRU. People always confusing us with Moru. So we are at the Faculty of Tropical Medicine, Mahidol University, and I would like to also thank the organizer for giving me opportunity to represent the establishment of the Vivax CHMI in endemic countries. So my talk today will be convened with the, when we need to establish the model, what to think about, especially when we are in the endemic countries, so many factors that we need to consider. So I classify into three parts, the one before we start the trials, during the trials, and after the trials. Before we start is the biggest challenge because with the ethical concerns, with the population engagement, with the university itself, we have to do some of the engagement to ensure that everyone understand what the CHMI is, and there are also the risks since even though Thailand is the endemic areas, but not everywhere in Thailand has malaria. So we are in Bangkok, in which we don't have malaria and no mosquito vectors, but we also have other diseases like dengue, Zika, chikungunya, which we need to concern. So the infrastructure and governance is also important to ensure that we can safely taking care of the volunteers, and also be able to optimize all the protocol up to the standard that required for the parasite banking and other vaccine trials in the future. And more important thing is that since even though vaccine believed to be benign, but we seem to see more and more severe Vivax malaria, especially in Thailand. We got a few death cases during the past few years. So clinical monitoring and medical experts on how to treat the severe malaria is also important. And another thing is the national ethical committees. So long story short, I think previously in Thailand, AFRIMS tried to establish the CHMI before, but failed because they cannot provide some of the information regarding the relapse, the safety of the volunteers, some kind of those things. Not because the trial is not good, but I think the data during that time not enough to show the EC committee. So we learned from other experience. So with this time we prepared, so we have all information on the epidemiology, whether it's drug resistant or not, and whether there's the chance of the relapse, whether the primaquine treatment is still safe to completely kill the liver stage hypnozoites. Those kind of information is very well incorporated when we engage with the ethics committees. And also the regulatory bodies. Ethics committee is one thing, but we also engage with the FDA even before we start the trials. So in Thailand, we don't have the law for biological research specifically, but we try to engage with them whether is there any concern if we doing the trial with the CHMI for Vivax, which will be further used for the vaccine trial in the future. But so far, they are pretty well aware, and if we follow several guidelines, the IND guidelines and other GMP compliance, those kind of things. And then during the trials, so with the trials, there are so many partners or the groups, like the one who handle the clinical team, the researcher, which also participate in terms of exploring the immunity or pathology, and also the data management team, which is also the big core group that people might not aware of. But with the clinical trials, there are a lot of process along the way. And one thing that I like to highlight for the trial management is that with the volunteer recruitments, and SAE is also one of the big concerns for our Vivax, because not only is the first one, but it's also in Southeast Asia, no one ever done that. Even though we get some primary data from the Angela that she mentioned earlier before. And with the data management, it's composed of whole list of the things that we need to do, but in overall it also need to be comply with the GCP, ICS GCP, and we have to have the independent statistician to not only analyze the data, but in some of the trial to randomize the challenging agent is also important. And at the end of the trial, so the trial with the patients end, but then we still have the further process like other clinical trials. But for VIVAX CHMI, one thing that we need to be maintained is the maintaining of the inocula that we produce under the trial. So with the current teams, we can do the monitoring, but with the next generation, because the challenge agent that we produce will be safe for longer, maybe after the next generation, so there need to be planned out on how to maintain the inoculum in the future in the proper manner. So for CHMI, the infection method we classify into 3 things. The first one is the natural routes of infection, which is mosquito bites. And the second one, the B and the C, is the man-made. The B is the sporozoid injection, in which you isolate the sporozoids and then inject to the volunteers. But this one is used very often for falciparum. But for VIVAC, so far we don't have the good cryopreserve reagents for VIVAX that can produce the reliable sporozoids. I know that Malaria may be able to do that, but so far in our hands, we prefer to go with the mosquito bite challenge. But also the weakness is that with the mosquito bite challenge or the sporozoid challenge, the volunteer can develop the hypnozoids. For the last one is the inoculation through the blood stage parasites, in which the volunteer will not have the relapse. And the endpoint is either could be the microscopy or qPCR. There are so many qPCR available in the publications, but we use our own established qPCR, which has been validated also with the public population. So in terms of establishment, we have to firstly start with the mosquito bite challenge, first thing first, because as Angela mentioned, we don't have the in vitro culture of VIVAX. So the only source to get infected blood is from the real patients. And then with that first mosquito bite, we generate the inocula, the blood stage inocula, which will be used further for blood stage vaccine trial. But anytime that you need to come back to pre-erythrocytic vaccine trial, you still need the patients and also the Anopheles colony to produce the infected mosquitoes. So we did the study at the faculty of the Hospital of the Tropical Diseases at our faculty. So we have the long experience on treatment of the malaria, both falciparum and VIVAX malaria, and with other capacity on producing the infected mosquitoes, very well established Anopheles dirus colonies, some of the epidemiology data that I mentioned earlier, the drug resistance, the relapse rate, the G6PD data, and highly experienced researcher, we decided that we do the study at the faculty in Bangkok, not in the endemic area yet. That is another plan. We have some concern on the transmission of the volunteers in the endemic areas. So the first one is the mosquito bites, which we call MIS, malaria infection study, MIS-1, to produce the PVIVAX inoculum blood bank, and then will be followed by MIS-2, the testing of the inocula that we produce. So since VIVAX cannot be cultured, and to get the infected mosquito, we need to go back to the field. And with the patients, you cannot predict when the patient will get sick and whether they will donate the blood or not. So it's more like the time matching. The volunteers that screen in Bangkok need to be matched well with the possibility that we get the VIVAX infected mosquito from the fields. So this is the example of the matching of the timeline. So with the trial sites, we need to screen multiple screening to ensure that we get qualified volunteers. At the same time, on the field site, during the desired time point, we do the membrane feeding, and then 2 weeks after that fed mosquito will be ready for challenge. And this is the whole list of the screening for volunteers with two main purposes. The first one for safety of the volunteers to ensure that there is no serious adverse events after the challenge. And the second one is also to save the trial ourselves. The key laboratory tests that we need to screen, and I like to highlight for mosquito bite challenge in this particular case, is the blood diseases, since this infected blood will be used for future vaccine trials. So we need to ensure that the blood of the volunteer is clean from other hematological diseases which might affect the ability of the parasite to grow inside the red cells. And also the G6PD and CYP2D6 genotyping to ensure that, number one, the volunteers can get safely primaquine treatment, and with the CYP2D6 to ensure that the primaquine will work in the normal metabolizers of the volunteers. And since it's also endemic population, even though not in Bangkok, we also do the test with malaria exposure to ensure that there is no previous exposure for malaria. So in this case, we use the established assays by our colleagues, Takafumi Suboi from Ehime University, in which we screen over 300,000 of falciparum and Vivax malaria antigen to ensure that these volunteers doesn't have any reactivity to those proteins. And then when the times come to the challenge, we also do other screening, mostly with the vector-borne and blood-borne disease, because in Bangkok we also have other mosquito-borne diseases. So we need to ensure that from the screening until the challenge, the volunteer doesn't expose with other infectious diseases. And on the right, pink box is the brief criteria that we use for our MIS-1, the eligible volunteers. And then when the time comes, we did the challenge with the 5 infectious mosquito bites. With this one in the previous, either in Oxford trial or Watery trial, is provide 100% attack rate. So we use this 5 infective mosquito bites challenge. And once by the volunteer, we monitor the blood parasitemia by the qPCR and also microscopic examination after day 5 to ensure that we can, number one, we can early detect of the parasite in the blood circulation, and number 2, for blood parasitemia, with the microscopic examination, we want to ensure that we really get the parasites in our inocula, because this will be used for further blood stage challenge trial. And the facility that we do that we process the inocula is dedicated for this study only, and no other study, so less worried about other compliance with the GCP and other things. And you might see that in Angela's slides already, that there's a whole bunch of the things that you need to do in terms of, facility preps and also the supply preps, those kind of things. And with the cryopreserve, we use the GMP grade glycerolide 57 for cryopreservation. This is pretty standard cryoprotectant for Plasmodium parasites. And once we bank, we also test the parasites that we banked with other tests, the sterility to ensure that there is no other bacterial contamination during processing, mycoplasma, bacterial endotoxin to comply with the IND, and also other blood-borne, vector-borne diseases, and also CMV, EBV, both viral and also seroconversion. And we also use our expertise on short-term in vitro culture to monitor the parasite in vitro, to kind of, monitor in vitro whether the parasites, after we donate from the volunteers, whether it can grow at least for 48 hours to be mature in the in vitro culture. And all of the inoculum agent was sent for genome sequencing. So far, we have 4 inocula produced by 3 different groups. The first one, number 001 and 002 is Vivax CSP210. I forgot to mention that in Thailand and maybe other countries like Myanmar or endemic area, there are two types of CSP210 and 247, which might impact the pre-erythrocytic vaccine efficacy test. So we also sequence this for every cases that we got. And luckily, CSP247 is very hard to find, but with the last 2 volunteers that we got, we were able to get the PV CSP247 sporozoid to challenge the parasite. So with the blood stage that we have now, we have both for 210 and 247. And some of them, the 001, 03, and 04 is clonal, but for 002 is the multi-clonal. And this would be the information for future blood stage challenge. And this is the example of the parasite dynamic in the blood circulation of the volunteers. Each different colors represent different donors. And the bottom graph is the malaria blood films. So overall, the qPCR will provide around 3 days earlier positivity for the blood stage parasitemia compared to the malaria blood film. We also did the membrane feeding for these volunteers to see whether is there any possibility to transmit to the Anopheles mosquitoes. But since the endpoint or the treatment the criteria to risk treatment for us is pretty early. For all volunteers that we have here, for MIS-1 mosquito bite challenge, none of them provided positive in the mosquito midgut. And this is a summary of the inocula that we got. Since each volunteers can correlate at different parasitemia, so that's why we end up getting variety of the blood parasitemia from the patients, and also the different number of vials that we frozen down. And so far, all the 4 volunteers complete the 1-year follow-up and no relapse of the disease, and no seroconversion for CMV and EBV. So then the next one is to test whether the blood stage that we banked can be also infective in another set of volunteers. So we perform MIS-2, which is the blood stage challenge for dose finding of inocula, because even though using the whole dose will possibly give you 100% infection, but that will result in running out of the inocula pretty quickly. So we try to optimize what would be the minimum dose to provide to the volunteer to still get the infective blood parasitemias. So for MIS-2, the screening is a little bit relaxed, because this one we don't need to worry about the primaquine treatment. So even though we test for G6PD, but some of the criteria we don't use as the exclusion criteria. We still maintain the list of the blood diseases, because as the outcome, it might impact the volunteers. So we still screening for blood diseases and also malaria exposure just for exploratory, not for the exclusion criteria. And with this MIS-2, we get better screening success rate 1 to 4 compared to MIS-1, which is 1 to 6. And then during the challenge is the randomized doses, which we try for 4 doses, the whole vial or neat, 1/5 of the dose, 1/10 and 1/20 of the dose. And with this one, we monitor the volunteers twice daily since the first day after the challenge by using the qPCR, the microscopic examination, and we also do the membrane feeding in this case, because one of the concerned if we move to the endemic population is that whether these volunteers will be able to transmit the disease in the areas, even though we have a lot of Vivax cases already, but in terms of the, if the volunteer happen to be the one that transmit, it will be ruin the, the trial, and even though it's not significant related to the incidence of the malaria, but it might not be good for the trial. So we did study the membrane feeding to ensure that whether we should do the trial in the endemic provinces or still keep the volunteers in Bangkok. And this is the data from each inocula. The upper one is the first inocula that we have, and followed by the second and the third one that you will see in the next slide. So for the first one, so for each group of the inocula, for each dose, we challenge with 2 volunteers. So for each inocula, there will be 8 volunteers in total. For the first inocula, of 8 volunteers came down with malaria regardless of the dose that they got. And the time to reach treatment is around 12 to 15 days. And the second inocula, number 002, we got only 7 out of 8 that positive for blood stage parasitemia. Now, there was 1 volunteer, the round green dot there, that didn't show reliable parasitemia except that time point. So we are now further investigating what might be the reason. This one, these volunteers, as you can see by the color, is 1/5 of the dose. So we pretty sure that the inocula is fine. But to further investigate whether it's the other underlying blood diseases or any other, dietary that the volunteers take during the trial. And very recently, we just complete the testing of the inocula number 3, the CSP247 one, which for this one, we also got 100% positive in blood parasitemia, which a pretty similar period of time to reach treatment in the volunteers. And with the membrane feeding for all the volunteers, since this is the dose finding, so each volunteer will not have the same or similar parasitemia. Not all of them provide the positive in the mosquitoes, but what I like to highlight here is that there is the possibility that volunteer might be able to transmit malaria in the areas. So this also helps us to design whether with other vaccine trial in the CHMI, whether we should do hospital-based or home-based, whether in Bangkok or in the malaria endemic area. And so far, this is the achievements of our study. So actually, the studies begin very early in 2017-18, but during that time, we haven't started any trial because we pay most of the time in engagement with the faculty, university, FDA, and also EC committees, not only the university EC committees, but national EC committees to get their, inputs on what's their perspective on CHMI before we actually decide whether we want to do the trial or not. And we got quite pretty useful feedbacks concerning regarding of the safety, how we handle the volunteers, the chance of relapse, the chance of drug resistance, those kind of things, which we have all the data to compile when we submit to the EC. So it's pretty smooth in the way that we have all the data ready in the package. And the first miss is actually start in 2020 and continue. This year we test most of the inocula to find the dose finding, and will be the last group of the mosquito bite challenge to produce the last 2 inocula stock. And we also plan to test the pre-erythrocytic vaccine trial, the RVX21 from Adrian Hughes group in 2025. Yes, and I think pretty much end. And yes, the last slide is the acknowledgement. So this is the first studies with the real collaboration between Mahidol Oxford Research Unit and the Faculty of Tropical Medicines. And you will see that it's composed of huge amount of people with different expertise, the researcher and also the clinical teams, and also the insectaries that make this study happen, and this study funded by Wellcome Trust. And that's end of my talk. Thank you.
Thank you so much, Mwalapa. That was a brilliant presentation. We have time for a question.
Thanks, Mwalapa. That was really great. We were looking forward for this presentation. And I thought that malaria vivax CHIM was complicated, but it seems way more complicated than I thought, especially with all the screening you have to do. And it's a very sophisticated and robust screening. And I'm wondering, what is your screening failure frequency? Because you have to exclude lots of things. So do you have an idea how many patients you have to screen to enroll one?
Yeah, so in terms of the volunteer part, for Miss 1 mosquito bite challenge, the success rate is 1 to 7 or 1 to 6, which means every 6 volunteers that we screen, we got only 1 that passed all the eligible criteria. For blood stage challenge, we get better. It's 1 to 4. Every 4 volunteers, we will get 1 eligible volunteers. In terms of the Plasmodium infected blood that we use to feed the mosquitoes, so far we got only 1 volunteers that happened to positive for HTLV. Others inocular that which excluded from the study. So others we get pretty much over 90% that most of the infected blood that we use to feed the mosquito will be cleaned.
That was great. Thank you so much, and I'm looking forward to having you in our discussions tomorrow about ethics and regulation. Thank you so much for your talk. So our last speaker before lunch is Dr. Rodney Ogwanda from Camry Unit in Kenya. He's going to talk to us today. Malaria? More malaria, yeah. Thank you so much.
Hi, good afternoon, everybody. It's a pleasure to speak to you today. I know we all we're expecting to see Melissa Kapulu, and hopefully I can be a good substitute. And since I'm the one between you and lunch, I'll try to be fast so that I don't stand between you and your food. My name is Rodney Ogwang, and I am currently a postdoc in Melissa Kapulu's group. And I'll be talking to you about how we've managed to leverage human infection studies in Kenya, and just highlight a few of the projects that we've been able to do on some of the data, so that we can spur. My understanding was to spur interest within Brazil for human infection studies. So hopefully I can be able to do that. So this is pretty old data from an old review from 2018 that just shows the different challenge models that have been put out, and the different infections that have been used. And you can see quite a large number of volunteers have participated. This was by 2018, so by now this number has grown quite substantially. A lot of the work that I'll present today is based off of malaria, Plasmodium, and a bit of work on Shigella that we've been trying to set up. So this just shows a map where different models have been done, human infection studies models in Africa. And you can see a lot of the work is mainly, a lot of it is, again, malaria, which is probably because it's one of the biggest diseases in Africa. But there's a lot of work. Moses will talk to you about setting up schisto. There's some streptococcus work that's going on. But to say that a lot of work is going on the African continent, which is exciting. So what have we been able to do in KEMRI? This just summarizes a lot of the projects that we have set up at KEMRI. The first one is to study acquisition of immunity and how immunity develops in adults. The next was a vaccine efficacy study. Then we've done a transmission block, tried to set up a transmission blocking model study. Then we've done some blood stage work as well, and then some Vivox work. And then we're currently setting up Shigella. For today, I'll only talk about the study of immunity in adults and present you some work on some vaccine efficacy studies that we've done, and just highlight a bit of the Shigella work that we are beginning to get going in KEMRI. So before I go deeper, I'll highlight Noni and Primus, who are going to talk to you later tomorrow, and they've done a lot of work. They've been super important in setting up any of the challenge studies that we've done, largely because they are the link to the community and the link to the ethics boards. And so when they speak, listen to them carefully, because without them, none of the studies would have really kicked off. So the rationale for infection studies, I think we all know understanding acquired immunity is one important rationale to accelerate vaccine development and to test the efficacy of interventions. So the next couple of slides will highlight what we've been able to do for these three different arms of use of infection studies. So the first, we'll start with what we've done with controlled human infection studies. It's been instrumental to be able to have a research grade material that you can use for infection. So Scenaria provided us with sporozoites, and then we're now currently trying to set up the blood stage infection model, which I showed you in the earlier slide, and getting material is through our collaborators in Australia. Right, so the first one, which is acquired immunity, we did what a controlled human infection study in semi-immune Kenyan adults. And the goal here was to understand acquired immunity in adults. And so we took 142 adults from 3 different sites. One in Ahero, which is a high exposure site, so a site with a lot of malaria. Then 2 sites in the coast in Kilifi. So one had semi-moderate exposure, and then the other site had low exposure. So these adults were infected with 3,200 sporozoites, and then they were followed up. And each had daily qPCR testing to look at the rate at which blood stage parasites were beginning to show up. And this was to correlate any previous exposure and previous immunity to the outcome of blood stage parasites. So this just shows what we saw, how the parasites were growing in the blood in the different arms of the study. So Nairobi is a low exposure site, and you can see in the low exposure site, a lot of the people developed parasitemia. Kilifi South is a moderate to high exposure site, and you can see in that population, a lot more people grew parasites to a detectable level, but did not reach the threshold for treatment. And you can again see the red dot that shows febrile episodes. There's a slightly higher percentage, but it's lower. Again, Ahero is an extremely high malaria transmission site, and you see again, there's a much more control of the parasite. So these can be summarized basically into this graph. What you see, if anyone is naturally infected with malaria, parasites will grow, and you'll see them in the blood growing exponentially, as has been shown in the previous talks. We saw interesting outcomes, again, susceptible individuals who, in the low transmission setting, parasites grow exponentially. We had a group of people that were slow growers, so parasites appear in the blood, but then these grow at a much slower rate. and did not reach the threshold for treatment and were treated at the end of the study. There's another group, are clearers, so the parasites appear in the blood, but somehow these individuals are able to control this parasitemia and actually clear parasites. So by the end of the study, their own immunity had cleared these parasites. And then we had this high group which were completely sterile, infected with a high dose of sporozoites, but the sporozoites, but the blood stage parasites do not appear, so they're called sterile. And so our goal now was to look at these different phenotypes and how can we understand the immune system, that the immune responses in these different groups of people to leverage what I told you about understanding vaccines, understanding correlates of protection, and so on and so forth. This is to highlight that this is a big undertaking with a lot of different groups at the site, a lot of people doing cellular work, others doing looking at different aspects of the parasite, and all of them are based at the Kemri Wellcome Trust site. A lot of people have been trained. I think there are over 15 PhDs that have come out of challenge studies, which is exciting for the continent. So this summarizes what I told you. So in the high transmission setting, you see that there's a lot more control of the parasite. High exposure, meaning high responses, meaning control of high immunity. Then if we categorize the people based on the anti-schizont antibodies, you see that these are antibodies to the whole parasite. And you see that people who have high levels of these antibodies are able to control the parasite. That's the graph on the right. So antibody responses are important for protection to malaria. What this doesn't show is which antibodies or which mechanisms of protection. So we undertook another study to try and identify which antibody mechanisms of protection are associated with protection. So this is a busy slide, but the first is a GIA, which is just basic neutralization assay. The next is an ADRB, which is a neutrophil-based assay. ADC is a complement. MOPA is phagocytosis by macrophages. Then phagocytosis by rings, and then the rest are NK-based assays. And simply what you see is antibody-based mechanisms of protection are a higher marker of immunity than just the basic neutralization assay. And when we classified, when we tried to group all the FC-mediated functions, you see that people who were protected have a higher breadth of the FC-mediated functions compared to non-immune individuals, suggesting that a breadth of FC-mediated functions is important, or an important correlate for protection. We've gone on to try and do some work identifying targets in those phenotypes, and this is work led by James Tuju, who expressed 111 immunogenic antigens and printed them on a chip, and has begun to use this chip to try and look at which antigens are important or important mediators of protection within the different phenotypes that we classified. This has gone on. I will not present any data, but a few antigens have been identified and are now going forward to kind of make new vaccine candidates. So to test the efficacy of interventions, I didn't really talk about the life cycle of the parasite because others have done that, but you can try to have interventions at the liver stage, which are anti-infection stage parasite interventions, or at the blood stage, which are anti-disease interventions, or at the transmission, so in the mosquito, which are transmission blocking interventions. And several interventions exist. Vaccines, monoclonal antibodies are coming to the fore, and drugs, and all these need to be tested in challenge models. So we've undertaken one vaccine study to try and test the efficacy of R21, which is a new candidate that it's been licensed now. So this is a bit old because the vaccine has already been licensed. But it's still, we had challenges because of COVID and because of our partners with Scenaria that had a few challenges with the FDA and providing some challenge grade material. But all the same, so it's divided into two phases. So the vaccination phase, each individual in the R21 arm received 3 jabs of the vaccine. And then we had an ME trap, which is a different vaccine. who received 2 jabs, and then there was a control arm. For the R21, we tested 2 routes of challenge. So one was ID and the other IV. While for the ME trap, we only tested IV, and then the control group. This shows the growth patterns of the parasite in the different arms. So the control arm, as you can see, everybody develops disease. For the ME trap, again, everybody develops disease. What was interesting was the R21 arm. Those who were challenged ID seemed to control the parasite compared to those who were challenged IV, suggesting that potentially there's a gap for the current R21 vaccine. So sometimes when the mosquito bites you, it bites directly into the vein as opposed to just depositing in the skin. So potentially a gap remaining in the R21 vaccine that has been licensed now. So I'll break here and say we're trying now also to set up the Shigella infection model. We'll not present any data as yet, but just highlight what we're trying to do to establish this. So we are currently still trying to get ethical approval. You mentioned the struggles with ethical approvals. It's been over a year that we're still trying to get the ethics to give us approval for the study. Hopefully we are close now, and then we will begin to do the experiment. So we'll challenge with S-SONI, and then establish the model, determine the dose, and then do a vaccine efficacy experiment. Again, I'll highlight this because without Noni, Primus, and team, it would have been impossible to get the study going. So there's been a lot of engagement ongoing with the ethical approvals, the regulators. So without them, again, nothing happens. So this is just a breakdown again of what we're doing. So we're going to establish the infection study model, utilize this to test the vaccine, then begin to do some work to determine immune correlates of protection and build regulatory approvals and frameworks for Shigella studies in Kenya. This is the initial study, which is a dose-finding study. The essence of this is to determine a dose that gives a 60% attack rate for all the participants that enroll. We'll do this in two phases. First, give 1,500, a dose of 1,500 CFU. Then after, if we don't get an attack rate of 60%, we'll repeat that with a higher dose until we find a dose that gives a 60% attack rate. This is because there's high exposure in the community, and so we need to find a dose that gives a 60% attack rate for the efficacy, for the vaccine efficacy study. So the next step, we'll do the vaccine efficacy study. We're still to identify the, anyway. So that'll be the next step. So in conclusion, I mean, community considerations and consultations in design is very important. Early engagement with ethical regulators is very important. And models are powerful tools for translation and discovery research. So thank you for listening to me, and thank you to all the partners that we've worked with, and mostly Melissa Kapulu for giving me the opportunity to present to you today.
Thank you so much, Rodney, for this brilliant presentation. Ricardo.
Great, very nice. For the R21 experiment that you gave IV and IM, right? If you compare both the antibody and the T cell in each schedule of vaccination, and what's the difference?
So that's still ongoing. The antibody levels look pretty much the same. The T cell work, we're still doing that in the lab right now, so I can't speak to that. But as for the antibody levels, they actually look the same. So it's something more to do with the route as opposed to a response in the blood as for antibodies.
One of the things I think you mentioned that really is important to stress is the huge opportunity for PhDs in development, career development of early scientists, that these kind of studies, particularly because they are multidisciplinary, skills required, can give to us. And I think I just want to emphasize that because, I think for a lot of us, particularly I can speak for myself, it was transformative for my career working in this field. And you had just mentioned 15 PhDs or something like that in this program in Kenya. So it's a really important aspect of these types of studies when we think about planning and doing that in low and middle income countries particularly.
Yeah, I'm a product of
as
the
well,
same,
yeah,
so
two products, yeah. Joshua is a product as well. Where's Josh? Yeah, Joshua is a product as well that I know of. So there is lots of us that have grown in this field and have done PhDs in these fields and postdocs, and I think it's a really important aspect to celebrate and emphasize. Great, thanks so much, everybody. We have lunch now. We are a little bit over the 15 minutes break, but we're going to bring it back on time. So if you can please come back here at 1:15 for the next session, that would be really great. And knowing, since you are in Minas, probably there is a little bit of food outside waiting for you, so enjoy.
What would you do if I sang out of tune? Would you stand